I'd like to introduce myself. My name is Alyssa Berger, and I am ATP-Bio's Administrative Director. I'm headquartered at the University of Minnesota. Today, you will hear from our director, John Bischoff, on ATP-Bio's State of the Center. And before we jump into the presentation, I'd just like to call attention to some upcoming public webinars. Tentatively, we have the next public webinar from ATP-Bio is tentatively scheduled of the State of the Research on Tuesday, February 13th. And we are also collaborating with the Society for Cryobiology on promoting their public webinar series. And so you can take note of these upcoming topics. Both of these webinar series do require registration, so watch out for some more information about those events. And for our academic community and our ATP-Bio member partners, we also have closed forums and additional events and updates from our community. So again, please watch for more information. This is just a heads up and an alert for you. We also have our upcoming Year 4 annual meeting hosted at Texas A&M in March. So I would like to turn it over to John for the State of the Center.
Great. Thank you, Alyssa. I'll go ahead and share my screen. Can everybody see my screen all right? Is that coming through all right?
Yeah.
Yes.
Okay. All right. Well, thank you all for joining for this State of the Center. And this is an opportunity for us to look forward and look back. We have had another successful site visit. There were a couple of pieces of feedback that I'm going to share with all of you that we're in the process of identifying. And we have a bright future, and I will just sort of dive right in. So I want to start by congratulating the staff for finding new ways to show our slides. And so all of the good new slides, that's basically Alyssa, Catherine, and the staff. And all of the old ones you can blame on me. So here we're looking at our societal impact slide in a new way. And we're focused on the same mission, but we're now breaking ourselves down into these three areas: healthcare, biodiversity, and food supply and sustainability. And then you can see emphasis areas within each one of those three buckets that we're focusing on as we move into the future here of ATP-Bio. This is actually something you'll hear this reflected, I think, in the State of the Center slides. And what I'm going to share with you is how can we do all of these things impactfully together? Where do we want to put our emphasis going forward so that we can actually achieve all of these goals and then bring this mission of preserving living biological systems for all to have societal impact. So one of the things that came out of the site visit was a request that we begin to think about our sustainability. And of course, we'll need to have a sustainability plan if we're going to be successful in our renewal. And the good news I have around that is that, well, there are two things. One is that we've actually started to draft a sustainability plan. But the second thing is that despite what we thought was the case during the site visit, that we would actually be doing our renewal next fall, that was actually incorrect. So Randy Duran, our new program officer, has told me that we are actually now 1 year out from that date. And so that gives us time to really put a robust sustainability plan in place. So you see also, this will be something done with the support of our Council of Deans and with obviously the leadership of our core institutions, as well as the main two headquarter institutions of Minnesota and MGH. So we're looking at further network engagement and governance, as well as obviously addressing our research, innovation, broader societal impact, and workforce development. And I won't read all of these specific aspects, but this is what we're going to be focused on in our sustainability plan, and this will all be shared with you. We've already begun sharing that with the deans, and it's being circulated amongst our institutions at the highest level. So to help us implement this, we have actually almost completely new cast of deans that are being led, of course, from the University of Minnesota. So Andrew Aline is our dean here, but we have new deans at MGH, Robert Kingston. We have Joe Elad at Texas A&M, and then Sujay at Cal Berkeley being assisted by Tarek Zodi. Only Chris Lynch was there for the original submission of ATP-Bio. So we're learning to work with this group, and they're learning to work with us, and they're here to support us. So this will be actually a big part of what I'm doing this spring, is actually going out to the core institutions and visiting with the deans and the leadership there to make sure that we have a firm and clear path forward as a center. So as I said, we've tried to freshen up the slides and deliver information in new ways. So I really appreciate this slide, which personalizes our org chart, if you will. So of course, you know Mehmet and I, but then there's also all of our pillar leads there, including our integration lead, Rhonda Franklin, and our ethics lead, Susan Wolf. And then our excellent staff members who are distributed amongst all of our pillars. And I won't call everyone out. But then also our thrust areas that you see on the right. And then EWD, DCI on the left there. So fantastic staff and leadership that allow ATP-Bio to keep moving forward. So one of the things that came up in the site visit, there were really two things, and one I've already touched on, and that is that they are looking for more active engagement of the Council of Deans, which is sort of to be expected since a lot of these deans are new to their position. And then the second piece was that we would show more leadership coming out of our core institutions, and not just coming out of the University of Minnesota and MGH. And so I've highlighted one, or rather two of our institutions, Texas A&M and Riverside. And what we're focused on here is, we need to have 3 core faculty and 3 students minimum at each of these core institutions. And we had originally, when we submitted the grant, UC Riverside was the core institution where Guillermo Aguilar was the head of the Department of Mechanical Engineering, and then he moved to Texas A&M, where he is now also the head there. And he has been effectively listed in both institutions. So we're looking at this spring and into the fall for opportunities to build up leadership and to actually find a third faculty, if you will, in both institutions, to bring them on board fully and to find true leadership for those institutions in ATP-Bio. And I'll say a few more words about that, because both of them, Texas A&M and Riverside, can really, I think, contribute to the biodiversity as well as the food and sustainability piece, as well as the healthcare piece of what we're trying to do in ATP-Bio. So we have recurring Zoom events, and this is one of them. So we are in the ATP-Bio public webinar series now, but we also have closed forums also shared on the Tuesday webinar timeframe. And we have Trainee Tuesdays, which is for any trainee in ATP-Bio. The closed forums tend to be more inwardly focused for ATP-Bio members. But the Get to Know ATP-Bio is generally open, and this is one example of that. This is an open event, and we have advertised this with the Society of Cryobiology, and beyond, of course, our ATP-Bio partners. And so we look forward to growing that community in these get to know ATP-Bio events. And I will also mention that we are also actively, as you probably saw, advertising the Society for Cryobiology webinars, which is also ongoing. All right, so of course, we want to continue to prioritize communications to all of our stakeholders as we build out, as I mentioned, the leadership, especially between the institutions, and making sure that we're connected between our trainees and faculty and more working groups. We just had a working group this afternoon on organoid cryopreservation, and I think we had participation across 3 institutions, which was really exciting. We have an intranet, which is for our academic community. We also have public-facing annual reports, and then we're on social media. We have both integration and ethics, as I mentioned, that are new to ATP-Bio, and I think potentially new to the NSF Gen 4 ERC program. These are not, at the moment, pillars per se, but they are areas of emphasis. And so integration is really important as we think about working across all these institutions that are dispersed across the nation, and keeping our projects clear and the communication clear amongst our groups. How do we integrate not just in the research, but also across all the other pillars to make sure that we're having our impact as an ERC? And so Rhonda, who is our integration director, sits over an integration committee that has representation from all of the institutions and functional areas. And she has they have an integration dashboard, which they're using to look to actually find areas where we maybe need to do a little more work to bring together, successfully our themes, and also to help everyone communicate ideally and build our community. So we're in our annual cadence. Of course, we had our site visit, which again, those of you who were there, I think you can appreciate that, at least for me, this is the first time I've heard of an ERC that didn't have to do overnight questions. That was thanks to amazing faculty and staff leadership and participation in the chat. We actually had Zoom going concurrently with live presentations. And so the site visit team was going back and forth with people. It seemed like at all moments of the day, there was a lot of activity there. And there was so much information shared that the site visit team really felt there was nothing further to be gained by having an overnight question, scenario. So that was great. And so now after that, we're basically into our new award year. And we're looking forward to the annual meeting, which will be coming up in Texas A&M in March, as you can see there. And then a little bit further on, we'll go into our data gathering and reporting, which will end up in our this coming year's annual report. And then finally, we'll repeat our ERC site visit in November 5th through 8th. And it again will be at the University of Minnesota, as it always is, in 2024. I just want to emphasize that, again, 2 of the things that came up in the site visit, one was more actively working with the Council of Deans so they can support the goals of sustainability in the ERC ATP-Bio, and then also finding the leadership role for each of our core institutions. And so I'm actually traveling outside of the annual meeting to Texas A&M and UC Riverside. You can see there's a leadership meeting there for UC Riverside in April. And that's specifically to meet with the faculty and the deans of those institutions to discuss exactly those points around the sustainability plan. And a couple of really exciting things around Texas A&M. we're inviting Nancy Curry-Grieg, who is the director of the Texas A&M Space Institute, to be at the annual meeting. And also I've reached out to Jeff Tomberlin, who's the director of an NSF IUCRC on environmental sustainability through insect farming, which I think will be really exciting. And I was just speaking with one of Shannon Tessier's trainees. He told me that they have a way of actually putting a black soldier fly larva into diapause at room temperature and leaving it there for 6 months, effectively metabolically inert, and then waking it up and having it continue. So there's a lot, I think, to be learned from working with these entomology groups as well as the space groups to focus on some of the bigger goals that we have in ATP-Bio, and I'm looking forward to that this spring. Again, calling out the annual meeting here, which will be coming up in March. The photos that you see there on the right are from our last annual meeting, so shout out to Thembian and the UC Berkeley group for hosting such a nice, wonderful meeting there at the Claremont Hotel, which was quite memorable. And then reminding you again that our site visit is coming up November 5th through 8th, 2024. So now I'm going to dig a little bit deeper into some of our pillars. You all know that Corkett and Catherine share the leadership of this pillar on the faculty and staff side. And Catherine has put this together in a more pleasing way to talk about some of the overarching barriers that we're trying to address: excessive ice formation, cryoprotectant toxicity, and thermal and mechanical stress. And we do this by convergence through let's call it hard science and engineering on the left and the medical and biological sciences on the right, bringing them together to actually work on these overarching challenges. So we're nationally diverse. That is, we're across the nation, and you can see that nicely depicted here in this graphic if you haven't seen that before. And the color coding here is indicating which of the three thrusts the particular lab it happens to be working on. And we've actually added some new folks here at the University of Minnesota, and I'll tell you more about our proposal, RFP process, and CERP process. This is a way for us to continually renew both our projects as well as our participants in the ATP-Bio. I think everyone has seen this, and I won't really say too much here, except that, again, to remind you, thrust one is around biological engineering, or preparing the living biological system to go into a state of suspended animation. Thrust two is multi-scale thermodynamics of water to actually achieve suspended animation, supercooled, partially frozen, or vitrified, or in an isochoric state. And then the third thrust is to bring the living biological system back in a rapid and uniform way that allows it to be viable and then to go on for some societal benefit or application. And that interacts with an innovation ecosystem that contains the other pillars, but also we have ethics and public policy, which again, I believe is unique, as is our integration committee to our ERC. Our technologies are really related to dropping the temperature in a sustainable way so that the biological system remains living. But as we drop that temperature, we drop the metabolism, and therefore we can store the living biological system for longer periods of time. The main technological areas where we focused are supercooling and partial freezing, which is primarily coming out of the MGH group, and that we call high sub-zero focus. And then we have lower temperature focus in terms of vitrified systems, which we at Minnesota, but also at Berkeley and Texas are also being explored. And the engineering piece of this, of course, is the manipulation of temperature, pressure, and the concentration of CPAs to depress the metabolism and reach this suspended animation. So what does this look like in a global sense? All of our thrust area projects, we actually have 29 core projects that are distributed in convergent research around the thrust, and you can see fairly nice distribution. What's interesting is that if you look at the overlap, you can see that there's actually a significant amount of overlap. At least 2 and sometimes up to 3 of the thrust are actually part of the core project. The yearly reporting associated with these projects takes the form of a quad chart, and then the information is then brought into the annual report, and is the site visit team and the NSF can look at this, but we can also use it internally. As we did just today we had a TA3 update, and we had something similar to the quad chart, which basically allowed us to very quickly and efficiently in an hour and a half go through all the projects that were part of TA3. And I think everyone agreed it was a very useful way to see in a broad sense all of the projects and look for overlaps and potential collaborative opportunities in the future. We also have a built-in evaluation process now that Corkett and Catherine have brought to bear, which is basically a red, yellow, or green stoplight, and with built-in sunsetting options for our projects. So we have the ability to add projects. We will also they will have a natural life course, and so these projects will also be dropping off and being renewed as would be expected. Associated projects, we actually have almost twice as many, 48, with $11.8 million in leveraged grants, which is about twice the average number for an ERC. We have system-wide test beds, and again, I really like the simplicity, but the impact of the new slide format. And so we can see with cells, focus on clinically relevant cells where cryopreservation really hasn't been very effective yet, and certainly has problems in terms of GMP and scale up for cell therapies. Working in tissue slices and microphysiological systems and organoids, cardiac, vascular, and pancreatic islets are 3 of the systems that we're working on. Whole organs, of course, heart, liver, limbs, vascularized composite allografts, as well as kidney, and then whole organisms. And you can see the examples there, zebrafish, as well as insects and shrimp, etc. So if we look back on year 3 in an overview, we can see that we've made some significant we've hit some significant milestones and achieved a lot in these different test beds. So we've increased RBC storage up to 160 days in supercooling, and Zia and Burke were the leaders on a PNAS position paper that was just published in PNAS, as I mentioned, around assessment of stored red blood cells using lab-on-a-chip technologies. And this is a way of increasing this precious, RBC system for use more appropriately and transfusing in the best possible way in a patient-specific manner, and using basically sensor technologies on the lab-on-a-chip to achieve that. In terms of MPS systems, 3D temperature-controlled cryoprinting, this was something coming out of Boris's group. And then in whole organs, we had the cryopreserved kidney that preserved the life of the rodent, which was a first coming out of the Minnesota group. And then ex vivo perfusion-based genetic modification that Corkett and the MGH group published. Actually, I'm not sure they've published yet, but it's a very exciting breakthrough where they're actually able to do the genetic modification in VCAs. We also have optimization of the cryomesh technology, which was used on a variety of organisms, coral larvae, Drosophila larvae, and even the much larger zebrafish larvae. We were able to show that we could vitrify all of them using the cryomesh, which had not been done before. And in many cases, we were able to rewarm them We were not able to rewarm the zebrafish yet, but we have some thoughts there. And then isochoric crab preservation and revival of Hawaiian stony corals. This is something Matt and the isochoric group did with Mary, which is very exciting. So as I mentioned, we have ways of bringing in new proposals, new talents, new ideas. And so there are two mechanisms for that. One is a request for proposals that comes out of the research program. And the second is a stakeholder-inspired research program that really comes out of our industry advisory board. And so you can see on the left and right, respectively, there, the RFP and the SERP, and then some of the topic areas that we focused on in the last round. So cell therapy products is an ongoing issue. Nobody that I know of has really addressed this, especially when you're dealing with 100 milliliter systems of these cell therapy products, CAR T cells, et cetera. And new approaches to organoids. This is an up and coming area of significant interest. There have been a lot of new NIH centers that were just announced in this area. And then scalable technologies for, of course, organ preservation. And then, preservation with an outlook towards biodiversity and food. This is something I'll come back to in a bit when we talk about the future. And then, excuse me, in the stakeholder-inspired research, which is, again, a little bit more translational focus, spatial and temporal measurement of CPA, high throughput production of CPAs and model systems, cryoprinting, molecular biology of the metabolism, volumetric current technologies, and advanced sensing. So there'll be another request coming out, I think, soon, and that's going to be on Corkett and Catherine to inform all of us of that. But this is, again, an excellent way for us to continue to move forward and remain creative and innovative. One thing I'll mention is that this year, because of what was raised in the site visit around looking for leadership coming out of the core institutions, all of these RFPs and SERPs that will be awarded only to leaders that are coming from our core institutions. So in other words, we won't be leading, Minnesota and MGH will not be leading any of these that will be funded this year. So I encourage people, especially at the core institutions, to start thinking about what they want to do in leadership, taking ATP-Bio forward in these different areas. So tangible impact of convergence on research. So one thing that I sat down, Mehmet and I, with Corkett and started looking at the original milestones that we had put together in the original grant. And we were kind of shocked in a really good way that we've achieved many of the milestones already in year 3. And we're getting really close to being done with these milestones. So it's really, it's amazing and very gratifying. There's also an increased number of interinstitutional and interpillar publications. So a lot of crosstalk in a really, really positive way, and an increased number of interinstitutional projects. And so one of our scientific advisory board members said, ATP-Bio pushes biopreservation light years ahead because of the cross-disciplinary and non-siloed nature of the center. And I think this is just another example of, what you can do and if you really have built a group that does convergent team science, which is what I think we are doing. And it's really quite satisfying to see. Publication trends are also very positive. I was just looking at this slide with Alyssa, and I think year 3 is actually cumulative, but even if it weren't cumulative, you would probably see it out here. So we continue to increase, and we are definitely above the average ERC. We're including multiple institutions, multiple disciplines, and our trainees are actively participating. And as you can see on the right, these are very high quality papers going into some of the very best journals that are around. So looking to year 4 and beyond, We want to focus now a bit more on biodiversity and food supply and sustainability. This is not to say that we're not interested in healthcare. It's just that if you look at where we've really already had so much impact, we have had that in healthcare, and we will continue to have impact in healthcare. But we want to, especially as we look for leadership coming out of the core institutions, we want to focus here in biodiversity and food supply and sustainability, where the core institutions can really help lead ATP-Bio into the future. So in innovation ecosystem, Tim, Carla, and Mehmet, of course, are leading this. We're engaging our stakeholders, connecting to our industry, and of course, they're supporting the center. And this is ultimately advancing innovation. And so we'll talk through a couple of key points here. One is the stakeholder-inspired research program. Again, we did this with the collaboration of our industry advisory board, and we've had 3 awards. So one is the microfluidic intracellular delivery of glass formers for neutrophil preservation. Prior to this, it's usually a physical process to get an impermeant glass former, usually a sugar, into a cell. And they've created a passive approach using shear stress, if I understand it properly, where the glass former can enter then in this passive way. And you can do this in a very high throughput manner. And you can see Nature Communications obviously appreciates this. And the paper has been accepted as of December. So that's a great win. There's also work going on in an all-in-one system for cryogenic transfer, transport, and storage at the clinical scale of vitrified human organs. And that's an invention disclosure has been submitted at Minnesota on that. And then a fabrication of next generation cryomesh for organism vitrification. This is a collaboration between Minnesota and UC Riverside, where Yedong Yin, who's an outstanding chemist, has looked at some of our initial ideas, and now he's trying to take it to the next level, maybe even taking it from 2 dimensions to 3 dimensions, which is extremely exciting. So the value proposition, of course, is that the ATP-Bio partners gain intelligence by seeing our research. They have the ability to influence us and help us, especially, for example, through the SERPs, to move in different directions. There's networking and talent going back and forth, and the ability to eventually hire students that are trained in ATP-Bio, and then ultimately we inform and engage one another. So we're extremely grateful to our ATP-Bio members. We are now 36 strong as of January, and this is a 33% growth in year 4. Many different types of ATP-Bio partners. You can see the industry are 22 sustaining full and associate members at the top there, but also we have nonprofit and government groups and innovation groups as well. And I think this really speaks to, again, these 3 areas of emphasis: healthcare, biodiversity, and food and sustainability, which is a broad bunch, I would say. And so, for instance, with biodiversity, this is going to be a different set of stakeholders that may not be able to put in the larger amount of money, but they can focus us on important problems and work with us with foundations to have impact in different ways. And this is an area, I think, that needs to have a lot more conversation going forward as we begin to diversify the IAB to go after those 3 important areas. We are starting to build a robust startup ecosystem, and these are some examples of startups that are working with ATP-Bio. And you can see on the right, we've boxed off the ones that I, at a first glance, just know that we have actually written SBIRs with. And you can see jointly over $14 million of SBIRs with these 5 startup groups alone. So membership growth goals, as I mentioned, these are the three areas: health, biodiversity, and food supply and sustainability. Health, I think, is doing well. We're collaborating. We have many associated projects, NIH funding and other funding, and engagement with lots of companies. I think we want to do the same now with biodiversity and food supply and sustainability. This is something that I'm personally taking forward into my visits to Texas and UC Riverside, and Mehmet is going to join me for at least some of those. And I encourage all of us to think about this. And even as you're listening to me speak about this, if you have ideas, if you can reach out, just send me or Alyssa a note, or Mehmet, about how we might be able to do this. I gave you an example of how we're reaching out in Texas to Jeff Tomberlin around, insects as food for potentially aquaculture. Also in Riverside, Riverside also has a highly ranked entomology program focused on insects that maybe are pest species to food. So if we can figure out ways to actually diminish the pest species, which is an ongoing idea that's quite similar to the one around mosquitoes, basically launching sterile males into a population to crash a wild population. These are all ideas that have cryopreservation as a core enabling technology that we can start to begin to think about. So in engineering workforce development, I'm sorry, I should have said, this is led by, of course, Keisha and Seth, both faculty and staff leaders who have really done a wonderful job moving us forward in this area, where we're inspiring, preparing, and advancing students and creating a real pipeline. So we're thinking from K through 12 all the way through undergraduate and graduate and beyond in terms of how we impact and prepare and work with students and trainees in this area. And you can see the leadership, both faculty and staff, spread across our institutions on the bottom. So we have both plan and execution in mind here. So strategic alignment and launching of new project review criteria is already being planned, and Seth and Keisha are launching that. And then we're planning going into Q3 and moving towards year 5 to build on our current momentum and invest in partnerships to increase impact, particularly around things like teaching fellows, as well as then moving to programs at scale. And that's where we really want to be as we go into our renewal, is to show that our programs are scaling and are having impact as we would hope. This is just some examples of what we're working on in our priorities, but also some nice photos of UC Riverside, the boot camp, I believe, from our REU program. Cal Berkeley there in the bottom right, they had a poster session where ATP-Bio was present. I think that's Guillermo with a design group at Texas A&M, if I'm not mistaken, and then MGH. I believe this was the trainee visit. The trainees had an SLC retreat at MGH. So we want to continue then to broaden access to undergraduate research opportunities, so REUs, but also beyond that, also in the academic year. Working to expand the senior design project, which you see, again, Guillermo's example here. We've done that also at Minnesota, and I think Purva has done that out in the Boston area. I think this is something that any mechanical engineering department could get interested in at any of our institutions, and then also building robust partnerships with community colleges, minority serving institutions, HBCUs, and beyond. So in DCI, Keisha again is our faculty lead, and Gina Rastani, our faculty lead, staff lead. And so we're working here to increase diversity and create a culture of inclusion throughout scientific discovery and innovation in bioengineering. And we do this through center-wide climate assessment, through our code of conduct and statement of diversity and inclusion, and then also capitalizing on new partnerships. And then I'll mention a few of the really unique things that Keisha has brought to bear in terms of DCI cafes, for instance, where we can engage in really important discussions around these critical issues and share as a community. a couple of examples here and also our priorities. Here you see Kevin Healy, PI at Berkeley, with Lisa Rogers, who Thembien, the faculty lead at Berkeley, helped to bring, and Keisha flew out to be part of this. Lisa was sharing from her experiences, I believe it's in chemistry, and that was a really big and important event out there in Berkeley with some ATP-Bio involvement. Here you see Rhonda with, I believe there's a Northside, I'm sorry, these are, yeah, LSAMP, I believe, REUs. Anyways, they are some of our REUs here in Minnesota. And you see our SLC trainees here at the site visit with Chris Hogan and Catherine. And then, of course, Keisha, who was delivering the pillar presentation at the site visit. So we want to continue to work with the LSAMP groups center-wide, and this is across different institutions, and I'll say more about that. The Lewis-Stoke Alliance for Minority Partnerships, strengthening partnerships in community colleges, MSIs, and HBCUs, and then a long-term action plan to address organizational culture. Here are a couple of examples. DCI cafes are, I think, the third Thursday of every month. It's coming up here on January 25th. And I encourage everybody, I typically go to these if I possibly can, and I've enjoyed every single one of them. The climate survey listening session will be on January 24th, and then implicit bias awareness on February 7th. And then you can see many other forums that we've had in the past that were also very impactful and useful to building our community. So finally, in ethics and public policy, this is led by Susan Wolf and Tim Pruitt. And as I mentioned, we are unique in having integrated ethics and public policy, as well as having an integrated integration committee. And so I'll tell you a little bit about some of this really impactful work, very exciting. If we look at what this actually is, there's really analysis around ethical, legal, and societal implications of our research. So that's ELSI, ethical, legal, and societal issues. And then advancing the ethical development and deployment of our breakthrough technologies. And finally, collaborating on engaging ATP-Bio and Gen 4 colleagues in ethics and public policy. And I'm going to give some examples of all of these things that Susan and Tim and their whole group have been highly effective on. And what they have actually done is to recruit a panel of nationally and internationally respected experts in the field of ethics and regulatory regulation of new technologies. And it's really exciting how they come together and have these discussions. And now they're moving towards actually disseminating some of these discussions to help really draw out a broader conversation about what we do in ATP-Bio and how what we do in ATP-Bio is going to go out for societal good. So a couple of highlights. We have added Shaniqua Collier to the EPP panel, the EP3 panel we call it. And there's a JLME, Journal of Law, Medicine, Ethics Symposium, which is turning into a volume of the journal. 12 articles are planning, will be published in the fall of 2024. Emerging Technologies to Stop Biological Time, the LC Challenges of Ethical, Legal, and Policy Challenges of Advanced Biopreservation. And this is, I've participated on a number of these conversations, and it is really a delight. Everybody who comes into those discussions, they're coming from a slightly different field or perspective, and you can't help but walk away with a new idea or an enrichment from that discussion. So these are going to be really exciting papers to read and disseminate in the community. In addition to that, Susan, with leadership from all of our pillars and within ATP-Bio, has really figured out a way to study ATP-Bio as a convergent team science group. How do you do this? How do you actually create, how do you build a large Gen 4 ERC, but then advance the training of responsible conduct when you're basically bringing entirely new ideas and technologies to bear. So this is using ATP-Bio as a petri dish, as she, I believe, once put it. There's also collaboration across a number of different papers, which is really exciting because that means we're not just putting out the research, but we're even thinking in the discussion section, even in the introduction, about how to position this correctly so that it's not just the research, but it also will be how it's accepted by and discussed by society. So that's really exciting to have that collaboration. We've had a number of seminars, one from Insoo Hyun around public engagement of ATP-Bio technologies, and he works at the Boston Museum of Science, and Gary Marchant around governance options for ATP-Bio technologies. And then I think in part because of what Susan has been doing as one of the leaders, the leader of this group, is that the NSF has really taken note. And so they now have formed a stakeholder engagement and impact collaborative, and she actually co-chairs that. So I think this idea of embracing and bringing in ethics and anticipatory governance and risk assessment, et cetera, into something like an ERC, I think that the time has come for this. And in particular for us, this has been really, really powerful. And Susan also invited us to participate within her consortium on law and values here at the University of Minnesota, which has been very useful to me personally. And Mehmet also, when he occasionally has shown up. And we get to have a group of peers, if you will, that are here at Minnesota that can give us advice, and we can share back and forth what's working well and maybe some things that we can learn from. So that's been excellent as well. Couple of accomplishments here, and this is really looking towards this Journal of Law, Medicine, and Ethics Symposium. There's going to be 3 sections, so they're thinking ahead in terms of, the top one being more on the healthcare side, the middle one, the bottom around conservation biology and food supply, the top 2 around essentially healthcare. And maybe the top one, I guess, is a little more general. But anyways, the point is that this volume, I think, is going to be really excellent and provides us a bit of a roadmap in terms of having these conversations around each of our 3 emphasis areas. And I'm super excited to participate in a small way, but also to read what's going to come out of this. So with that, I want to thank you all for showing up. I think we have a couple of minutes here, and I will stop sharing and be really happy to entertain any questions that you might have.
Thank you, John. And like John mentioned, we do have some time for open question and answer. You're welcome to unmute yourself and ask John a question directly, and that up to any of our ATP-Bio leaders. Or you can leave a question or comment in the chat, and we'll respond to that as well.
Well, maybe I'm going to pick on people if you're not going to ask me questions.
John?
Yes.
This is Mesh. It was a great presentation. Just one comment, a small comment.
Yep.
In one of the slides, you say research is education, which some people might find to be somewhat inaccurate or not totally correct. I mean, they're intertwined, but research itself doesn't necessarily have to have education as a part. So I don't know if you want to sort of discuss that, or, I don't know if that's what the NSF is looking for or what.
Yeah. Well, I think that maybe a more careful wording is something I'd be happy to look at, and I can run that by you if you like. But sure. I think the concept is simply that, we're convergent in the sense that one thing informs the other. So I think the concept was really that as we do research, it can be an opportunity to also learn and educate. I think that's really what we were shooting for there.
It's an important concept that the way NSF has funded research is by training the next generation. In other words, they had the opportunity to just take, experts and give them money and have them do research, but they chose this particular platform of at the same time training the next generation. So, but so I think it's a valid notion, but maybe the wording can be slightly changed.
I take your point, and I will just reflect on a meeting we had with Deborah Jackson, who's part of this ERC. I think she was there at the inception, and she pointed out that ERCs were originally created because people in industry were complaining to the government and NSF that the students that come out of our programs are not well trained to communicate and actually do, research in the corporate world and industry. And so the ERC in some sense was created around the concept of doing a better job educating.
I think she's probably altering history. I was there. and that wasn't the reason. It was a Japanese competitive initiative. But everybody can rewrite history the way they want.
Okay. Thanks, Mesh.
You bet.
I don't know, Susan, do you want to, did I cover your JLME symposium? Did you want to say a few more words?
That the articles are due today.
Yeah, that.
They are pouring in and they're thrilling. And almost all of these articles represent collaboration across ethics and the science and engineering and ATP-Bio. And the guest editors are myself, Tim Pruitt, and Corkett. So we are really, this is not as powerful as I think this collection is going to be on ethics. It is much broader and much more collaborative. So stay tuned.
Fantastic. Yeah, and I also wanted to call out, I don't know if he wants to unmute and say a word, but our Dean, Andrew Lean, is with us. And I think this is really great that he's here. And I don't know, Andrew, whether you caught what I was saying, but we're really looking forward to working with you and the Council of Deans to get support and advice on our sustainability plan.
Yeah, thanks, John. As am I. I sort of, again, look forward to working with you on that. I think that's one of the key agenda items that I have associated with ATP-Bio for this upcoming Q1. I will, right after this, and you'll note, to see if we've closed the loop on the final version of that sustainability plan that we can then start distributing to the other deans. I'm already starting to get some traction from them. So to other folks are on the line too. Good idea to let your deans know what's going on in a concise way, perhaps packaging it all up and then just, quarterly, just, hey, here's what's going on this campus. They'll hear about it multiple ways. But if you could put that into no more than a page, that would actually be pretty useful for the rest of the Council of Deans, but I'm sure as well for John as well too. But yeah, I was here for the presentation, so thank you very much for the edification. It's good. Thank you.
Thank you.
Thanks, Andrew. All right.
And it looks like we have a question from the Minnesota group that's in person in our ATP-Bio headquarters space.
Yeah, right on. Thank you. It's Nick Zahovich here, and we've got about a dozen people here, eager trainees packing the room in the new ATP-Bio conference space. So I'm delighted to see the increased emphasis on biodiversity, very much of interest to me. And I'd like to just ask, how do you envision that being implemented in practice? Does that mean more faculty? Does that mean, finding more grants to pursue? Where's that going?
Yeah, it means I have to reach out to people like Susanna Williams and ask how we can better collaborate with some of the ATP-Bio partners in this space. Hi, Susanna.
Hi, John. That was great. It was great to see you guys and hear such an update. So the question was how to collaborate better with people that are not in ATP-Bio?
Well, within the biodiversity space, Nick was just saying that he was delighted to hear that I'm making at least I'm giving some lip service at least to
Well,
biodiversity. So
yeah, as someone who is obviously working in rhino conservation and other species, and actually I'm currently just writing a protocol on how we can work out ways to cryopreserved insects, butterflies, which of course it doesn't happen at all. I'm literally writing it now. And it's been given to an incredibly high important individual in London next week, which I can't say who it is, but anyway, it doesn't matter. You're all very important, but you get the drift. Anyway, so I think my question is the biodiversity is someone who's external to the USA. I love your beautiful map of everyone in the USA. Is there any way I can be involved or partners across the world?
absolutely. Catherine and Alyssa, I don't know if you caught this, but they are making new slides. They would love to include Europe and the rest of the world. In fact, Catherine's not with us in the United States today either. So what do you think, Catherine? Can you do that?
I can do that, yes. I'll reach
Wonderful.
out.
All right.
And
I look
moreover-
forward to that, Susanna. Let's do that.
And
Fantastic.
moreover, we
That's good,
just
John.
brought Taronga Zoo on, so we've reached out into Australia. We can certainly reach out elsewhere.
Excellent.
Fantastic. Great. Well, we have a couple more minutes. If anybody would like to give an update or if any thoughts that I might have triggered, anybody feeling good about food and sustainability?
John, can I just add in response to Susanna's point, you may be interested to know that in our outstanding group of external ethics and law consultants, we have Evelyn Brister, who is a philosopher of conservation biology, Paul Thompson at Michigan State, who's probably the country's foremost philosopher of food and ag. And so I'm probably missing someone. We have also Andrew Menard, who is a leader in responsible innovation quite broadly. I thought of him because that's an approach more identified with the UK and Europe, but he's really done a great deal. He's now at ASU to bring it across. So we are thinking about some of these things also already in the ethics and public policy piece.
Fantastic. Thank you. Thank you, Susan. And Guillermo, did you want to take the last minute and just, welcome everyone to come down to Tamu?
Sure. Well, I look forward to hosting all of you here in Texas once our really bad weather here goes away. Today we're at 29 Fahrenheit, John, and we're freezing here.
Oh, come on. We're at like 7. Don't give me a.
I knew you would protest. So, no. Anyway, March will be a beautiful time of the year over here. So I'm really looking forward to welcoming you all on board. And I see Matt Powell-Palm joined the meeting as well, and he's going to be one of the hosts as well, whether he knows it or not. Yes. So anyway, that's great. I'm really starting to like the way this center is integrating and we're collaborating across the different core institutions and everyone else who wants to join.
Great. Well, Alyssa, you want to send us off?
Sure, yes. Well, thank you again, John, and thank you to all of you who joined our first public webinar series. And we'll share with you what's coming up next. Look for some communications and promotions about our upcoming events.
Thank you, everybody.
Thank you.
Take care.